It's a really, really exciting pleasure to be giving the Grand Challenge today. I'm delighted and thank you for the invitation. I am going to be using you guys, though, as guinea pigs. The work I'm presenting is all work in progress, and I'm actually going to try a script today for the first time, because that's how I usually write scripts for my animations right at the very, very last steps. And so it means I'm right at the last steps of this particular production. So I'm not sure how well it's going to go, but we'll give it a shot anyway. So I'll do a live narration that will be recorded at a later stage. So to frame the grand challenge, I guess, of what my work is addressing, I think it's really tackling how we perceive ourselves, how we understand ourselves to be what it is to be a living being, to be alive, to be a living chemistry. And that's what my work tends to do. It drills down and shows us the mechanisms inside our cells and how we work. All biological life, everything, all life on Earth depends on the flow of energy, and that's what we're going to focus on today. So the activity inside your cells is all powered by converting the food you eat and the air you breathe and converting that into chemical energy. And that very specific topic is what I'm going to be looking at and showing you today. So actually, I'm going to turn down the lights because I certainly prefer it that way. I'm fussy that way, having spent so long getting the colors just right, and then to see them washed out on the screen depresses me. Let's find page one. So this is the location where we're going to be spending our time in the next half hour or so. This is a beautiful microscopy of a living cell. The purple is the surface, the membrane, the outer surface of the cell. The big blue object is the nucleus with all of the DNA. What we're going to be focusing on is the yellow objects. These are mitochondria. These are commonly referred to as the power plants of the cells, the power generators of the cells. In textbooks, classically, they're usually illustrated as these little jelly bean objects, where, as you can see, they're very dynamic and they're constantly branching and fusing and all sorts of dynamic behaviors because they're constantly responding to the needs of the cell, changing needs of the cell. So the yellow objects, the mitochondria, are the landscapes. We're going to go inside those and we're going to see reconstructions of what it's like inside of the mitochondria. And this is the molecule we're going to focus on. It's adenosine triphosphate, ATP. All life on Earth, no exceptions, all life on Earth use this to power themselves. It's the universal energy source or the currency of living cells, all types of life on Earth. It's also a precursor for making DNA and RNA. So it's really the key, or a key molecule for all life on Earth. It's key for origins of life stories as well. And so this is what we're going to focus on. Okay, so right at this moment, your body contains about 10 to 15 grams of ATP. Every second, your body consumes 1 gram of ATP. So in a day, you will recycle or reuse over your body's weight in ATP every single day. So the question is, where does all this ATP come from? Deep inside your cells, rows of molecular motors are creating ATP, or recycling ATP. The molecule that's essential for all life on Earth. ATP is generated by a rotary molecular motor, or molecular mechanism, enzyme synthesis of ATP. Chemical reactions are pressed together to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP in each cycle. Inside the molecular motor, rotating axle powers the sequence. Powered by the force of protons moving through the rotary motor, ATP is generated. A difference in proton concentration on either side of the membrane, powering the force pushing that rotary motor. ATP drives most the biochemical activity inside the living cell, and it's a key building block of DNA and RNA. So this particular animation is meant to be a scene setting for students to introduce them to the place and location, the context of ATP synthesis. Everything I'm going to show you is all derived from various forms of data, and I'm going to break this animation apart and show you some of the data that went into informing how this would end up looking. So I'll show you the different components, and then we'll revisit this animation so you can see it in context. So this is a paper, a review paper that came out in January this year, so it doesn't get much fresher than this. And it is of the ATP synthase mechanism and a review of its structure. These are just representing different representations of the ATP synthase. So this is a ribbon diagram. It's very informative for molecular biologists to see it in this format, to see its underlying structure. And this is an animation that produced by the publishers, the authors of this paper, to show the rotary mechanism of ATP synthase. And then these are other forms of data. So this is the model. So this is a rotating model, and it's originally, they're using tomography, but they're also using X-ray crystallography. And that's always that's the first place, is to get the model, the 3D model. And this is unusual to be able to have a representation of how it operates published in a paper. This is my representation. So using that data and then transforming it into an animated model. I prefer the sort of surface van der Waals type representation. It gives you really much more, I think, a sense of the 3D form, especially if you're non-expert in molecules. And what I'm demonstrating here is the reactants coming in and the overall operation of ATP synthase. But this turned down the wrong mover. Quite so much. So what we're focusing on here is one of the proteins, one of the enzymes within the system. It's a jelly bean-shaped enzyme, and it's being opened and closed. It opens up, and then the reactants will randomly come in. But once both reactants, the phosphate and ADP are there, it then will force them together to form that chemical bond. And this is the rotating axle with that rotary motor. In this case, in most cases, it's usually protons that are driving that sort of water wheel mechanism. And certainly in all of our cells, it's proteins that are doing it. But again, this is all structural data that I've been animated and brought to life. So when I showed you the earlier animation, it had these pairs of ATP synthase. This is some of the data that went into why I reconstructed in that way. And this is where they're looking at using high-powered electron microscopes. And you can see the pair of ATP synthase, of these silhouettes here. And where the red is, that line is where the membrane is. And you can see the membrane is actually being held under tension in this sort of a shape. And the two ATP synthase are locked together. And we'll come back to that reason why it's doing that in a moment. But all of these measurements, looking down on the ATP synthase, these precise measurements, all of that goes into informing how I'm going to build the visualization. And this is where they're overlaying X-ray crystallography, precise atomic detail, with tomography, 3D reconstructions, two different forms of data with different kinds of capabilities to reconstruct what the ATP synthase is like inside this particular type of cell. So this is my simple representation, taking X-ray crystallography and animating it to show the pairs of ATP synthase working together. We have a membrane here. We have a membrane here. This is actually outside of the interior of mitochondria. This is actually interior of mitochondria. So like sort of pipes that run through, membrane pipes that run through the interior of mitochondria. Much higher concentration of protons on this side, that side. That motive force of the protons is the rotary motor that is driving the mechanical mechanism to make ATP. Next bit of data, these are now looking at the rows, these pairs of ATP synthase, they're forming rows. These are different organisms. You have a cow on this end, looking at the shape, the angles of ATP synthase, and then these are electron micrographs are looking down on top of these, how these pairs are now forming rows along the interior membrane of the mitochondria. These are 3 different types of fungus and yeast. And then we have potato. So these little, you can see the sort of how potato has these very wide ATP synthase, and you can see there's a lot of variation and variety in them. And these are only looking at this system where it's actually manufacturing ATP. There's this ATP synthase, there's many different variants of it, and in fact, ones that run the opposite direction that will use ATP to turn that turbine the opposite direction and pump protons in. And that, for example, is in your vesicles inside your cells where you use to digest things. It'll actually have ATP synthase, or the opposite, hydrolysis, where it's actually pumping protons in, creating a highly acidic environment to break things down inside that. But it's essentially the same mechanism, but repurposed for a different use. So this is a very simple visualization, looking down the top of those pairs of ATP synthase, all the measurements needed to reconstruct this visualization. And so I'll play this animation again. This is what I'm calling the cinematic. It's meant to engage students. The sound design is all the sound, in fact, everything you're seeing here is all work in progress. But the sound design, actually, I'll come back to that in a moment, but the choir or the singing you're hearing is just a placeholder. We're actually working with a school in Melbourne, and their senior choir is going to be singing this for us. And we're also going to have one of the schoolgirls is going to do the narration, which I'm really delighted about because these animations are commissioned primarily for the US, and I'm really happy that it'll be a schoolgirl Australian voice that will actually be the international version for all of these animations. Also, I'll point out that all the color palettes are me currently adapting to having color palettes that are appropriate for colorblind people. Because 8% of males are colorblind. So I'm using yellows and blues actually as contrast, but I usually use pinks and reds. I always have, because it's very evocative of something that's alive and healthy. We always respond to pinks and reds that way because we're mammals, and we like that. And certainly, if I went for pure blues and yellows, it would make it look much more for people who are full spectrum color, it would certainly look more cold and lifeless, I think. I usually use blues for death or dead things or things about to die. Yellows and greens are also disease colors. So it just evokes a feeling. But in this case, I'm using pinks and reds to make it come alive. The blues and yellows are there for contrast, and it's part of the consideration of crafting this for all sorts of audiences. But I'll play it again. I won't speak over the top of it this time so you can see the context of it. But this is the foundation for a whole suite of animations and interactives and all sorts of things we're doing for school kids and school groups and university students. and all of my work has always been driven primarily by the games industry. Well, it used to be Hollywood. Hollywood was doing, with special effects, was driving computer graphics in the early 2000s. But now, well, in the 90s, and certainly now, it's clearly video games, because they can certainly achieve cinematic quality equivalent to Hollywood, but now it's all in video games. And what I'm actually playing you here, it's been commissioned by the Howard Hughes Medical Institute in the US, and they want animations, videos, working with them a long time, because it gives a lot of access to teachers in the classroom and to students. But this is now all developed with a game engine pipeline where this isn't actually a movie. This is all live all the time. We're getting 4K, 30 frames a second, solid, where all of these things, where each of these ATP synthase has 100,000 atoms. We now have, 300 or 400 ATP synthase there. We have membranes, which used to be the hardest thing to do in animation. Now it's just like a click and it's done. It really is. We're going through a really radical transformation now in the last 2 years. And not only that, so we can create movies now, and we are doing 4K movies, we're doing 8K, 360 movies, all sorts of things. But I'm more interested in actually having it as an interactive live world, particularly on mobile phones. I think that is actually the key for access for students. And one where they don't actually just watch the movies, it's where you can fly to any of these ATP synthase and you go look at it, you can go around it and interrogate it. And every single one of them are all fully operational and have all the detail there. It's more about just developing a user interface that's going to make it accessible and usable for students. But that's been a really tremendous transformation for us. It was originally driven by the need to do, we're working on dome films for planetarium-type shows or science centers with giant screens. And this would start in 2014, and we needed to do a lot of high-resolution stuff, and it used to just be the biggest gut ache to try and render, and amount of time, months spent waiting for machines to process imagery. Now, that's completely gone now. It's all about building detail into those worlds and setting up storytelling through camera setting, but it's not about hand-wringing, waiting for the machines to render. You wait 10 days and go, that's not what I wanted, and wait another 10 days and render it out. It looks crap. And like that used to be my routine for years and years and years, and now that's just changed. It's profound. But not only that, it's also the nature of how the audience is experiencing these things. It's not a movie, it's an interactive, or it can be. For example, this is we're just dabbling in it, augmented reality. This is exactly the same model as you saw before, the ATP synthase, and it's effortlessly playing. We have 100,000 atoms, fully animated, seamless loop, and it's now in augmented reality easily. And I mean, this is just what I'm really keen for. This is every kid has a phone in their pocket. Give it to them that way. They look through their phones, and on the tables in ATP synthase, they stand around in a classroom, and I think that's where they interact. And I think that's clearly the future. So we're just dabbling in this. I'm getting very interested in it. I'm certainly, having come from more of a cinematography type production, that's certainly where my craft is, and that's where I care about. But I clearly see the potential in this, or this is what audiences will expect. It's not a matter of whether we do it or not. We have to do it. It really is profound. So this ATP synthase actually operating at real time. This is how fast it operates inside your cell. You have hundreds of thousands of these in each one of your mitochondria. This is about how fast they're operating. Transformed somewhat so a human audience can understand it. But so at least you see the speed of rotation of the motor, and it's producing, in this particular visualization, 80 ATP per second. It's probably 30 to 50 per second is a typical because of the constraints of the cell and the supply of components. And in bacteria, it's expected to be about 100 per second. And the reason I'm playing that is, again, we'll go back to, this is the world, this is the location of where these visualizations are happening. We have our cell. Inside those mitochondria is where this stuff is happening. And I'm going to show you visualizations reconstructing the interior of the mitochondria, but everything I'm playing now, the mechanisms are now operating in real time, in quotes. It's all been adapted so that we can watch it. It's certainly to show the true speed of molecules and how they'd be dissipating or Brownian motion would make it so confusing we wouldn't be able to watch it. So I have to adjust it and make it meaningful for an audience. But as much as I can, I will certainly retain real-time speed, things like that. These are structures are accurate for size and how they relate to each other. I think that's key. But I have to adapt it and make it stories. One last bit of data before I show you some of the reconstructions. This is a beautiful piece of tomography from Sweden. So I showed you the mitochondria, these little wiggly worms just before. This is like when you slice through a cucumber. This is a slice through a mitochondria done by tomography, which you guys do beautifully here. But this is a huge slice through a single mitochondria. And slice by slice by slice, they've reconstructed the 3D shape. And these are just the membranes, the tubular network I was describing earlier. These so to show you the complicated tubular landscape that's inside mitochondria. And so those ATP synthase, the visualization I showed would be a tiny little spot like that before. And what we want to do is reconstruct this kind of the whole world, particularly for dome experiences or virtual reality where you're looking around. You want to be able to be placed inside this world and be able to look around and experience it all. So this is what I'm reconstructing. Just an aside, those blue blobs there are the genome of the mitochondria, the 13 genes and other information inside the DNA of the mitochondria. That too we'll be coming back to hopefully next year, actually, with replication. These are, from middle of last year, these are reconstructions of those membrane surfaces, those tubular network inside the mitochondria. And we have ATP synthase, those pairs running along the edges, and they're creating that fold, that bend under tension of the membrane. I won't go into that now. But then we also have on the upper surfaces, on the flat surface, or more flat surfaces, all of these other proteins. And they are part of they were all responsible for taking those protons from one side and pumping them across the gradient and putting them on the other side. So there's a high concentration one side to the other that ATP synthase then can use to power its synthesis. So now we're going to look at a few of these mechanisms, or only one of these mechanisms, unfortunately, today, and look at how this all comes about. This is all respiration. This is classic 101 biology that high school kids will do in their final years, or university students certainly would in biology, is looking at these kind of mechanisms. And this is the best figure I've seen it's actually from a review paper, a published literature, but I think it's better than certainly the high school textbook illustrations I've seen. On the right-hand side here, which way are you guys? Yeah, right-hand side here is ATP synthase, so that illustration of the molecule you just seen. And that's known also as complex 5. There are 4 other complexes in the membrane, embedded in the membrane. All the other 3 are to do with the breakdown of the food you eat and extracting energy from that, and then using that to pump protons across the membrane. And so just that one mechanism, this is a classic representation of it. It just has a few arrows saying ADP and phosphate go in, and ATP comes out. And there you go. And that's it. This is higher. This is better quality than I would see in most textbooks. But that's kind of the level that students would study. Where I think you have to see a dynamic to really fully understand what's going on and watch it in action. And what we're going to do is just focus on just one, unfortunately, because this is all work in progress, and I'm not allowed to show you much more of this current project, but I'll at least give you a taste of what's coming. All this will be published in probably the next 1 or 2 months, and it will be on various websites and so on. So all these materials are available for free. We provide it all around the world to museums, science centers, but primarily for classroom education, and translated to lots of languages and so on. But that's sort of the nature of what I've been doing. So anyway, we'll come back to. So we have complex 1, 2, 3, 4, and 5. We're just going to go to complex 1, the very beginning. This is the first step embedded in the membrane inside the mitochondria of taking those electrons from the food you eat and using that to put it to work to pump protons. And I'll show you the mechanism for that. Oh, okay. So one last thing before we head into that. Again, we're back to this tomography. And this is a relatively thick slice through the mitochondria to show the membranes. But you notice that where it cuts through those tubular membranes, that's what these little representations here are like. And that's what I'm going to show you in a moment. I'm going to reconstruct as if you had done a slice through the tubular membrane network, and we're going to look down one of those. So Here we are sliced through the tubular mitochondria, internal membrane of the mitochondria. We have those pairs of Atp Synthase on either end, one there, one there. And the quality, I haven't seen any confirmation in the literature, but I've seen it mentioned twice. The quality is why it's holding it in that kind of a membrane, is creating that sort of under tension. Why it bothers to do that is it actually creates a slight negative charge here, which attracts the protons that are positive to gather. It's like an efficiency. It actually improves the efficiency of how many of the protons that are available for driving the rotary mechanism of ATP synthase. So what we have arrayed around, we have the two ends, we have ATP synthase. Just here we have complex 1. That's complex 3, 4, 2. And in the interactive, students will click on them and then zoom in and explore them and so on. So, thank you. So, we're going to go to complex 1 today, unfortunately. This is a reconstruction of how complex 1 works. This is the membrane of the mitochondria here. This is actually the interior of the mitochondria. The exterior here. This particular complex, enzyme complex, is embedded in the membrane. And what it's doing is taking, when you eat food, the sugars and the proteins and so on, or sugars are broken down, one of the steps is this little courier molecule actually takes electrons from that system, a breakdown of food, and it's carrying in those electrons. It's dropping them off here, and the electrons are actually passed to the interior of the protein down to this other courier molecule called coenzyme Q. It lives in the membrane. It lives vertically in the membrane. It's able to hold the electrons. So 2 electrons dropped off here. They pop all the way down through. They're passed through the middle of the enzyme, and then they're passed to the courier molecule here. When it does that, the charge running through this protein causes it to flip and fold forward, which we're not quite sure all the details, but causes 4 protons to then come through. 1, 2, 3, 4. And they're being pumped through. So electrons are being received, they're being passed through, and that's being turned into mechanical pumping of protons through the membrane. One last time, this is all playing in real time on our gaming, just like a normal gaming PC that every teenager would love to have in their bedroom. It's now playing live, but at the same time I still love my movie making, and these are fabulous, that kind of stuff. But anyway, that unfortunately is as much as I'm going to be able to show you of this particular project, and I'm really disappointed by that because further upstream from this is all the very final steps of metabolism and respiration, breaking down the food and using air and oxygen that you breathe to make energy. But further up the stream is something called the citric acid cycle, which is really the hub of all life on Earth. It's where protein precursors are made, lipids are made from it, sugars can be sort of made from it. It's essentially the engine, the rotary, a chemical cycle that produces all the constituents for you to make your body, to make all your living, all life on Earth produces from the citric acid cycle. And it's also diabolically complicated chemistry that students have to learn all these complicated terms, and it's just a memorizing process of arrow flows and all these chemicals. I never understood it. And I went through and doing PhD and whatever in biology. It's awful. But when you actually look at the actual, I've gone through the step-by-step through this 8-9-step process. Every single one of those steps is a fantastic, awesome molecular mechanism. And there's 2 in particular that are just going to, I think it's just incredible. I mean, really, when to see it, it's going to be astonishing. And then before that is actually glycolysis, which is another 10 steps. And I've just had a look at that. They'll be done, finished by the end of this year. But those 10 steps, again, every single step, of course, it's an amazing little mechanism. It'll make sense to see it in action, to see it dynamically. And I think it'll be memorable just of its own accord, not having to require it to be memorized, per se. All of my work, or the project I just showed you, I I'm based at the Walter and Eliza Hall Institute of Medical Research in Melbourne, and they have supported me since 1995, and so I'm very, very happily ensconced there. The Howard Hughes Medical Institute is a major philanthropy. They fund tons of medical research all around the world, and they have been an ongoing support of my work. I've done multiple projects with them, and they're a delight to work with. And just recently, I've the Telematics Trust, which are based in Melbourne. It's my first philanthropy that's actually supporting my first Australian philanthropy that's supporting my work. And they have supported the augmented reality development, a lot of, and also all the sound design that we're currently doing, which has enabled us to go to a school choir and we're doing lots of different things. And also to develop a sound library that will work with interactive works. So we can place different sounds with different objects so that for engagement and for storytelling, it's going to be a long process, but it's going to be a very interesting one. So this is who's supported that work you just saw. And they sort of my key supporters over the last decade. And just to finish up with the credits for this current particular work, this is my team. It's me. Justin Muir joined us 2.5 years ago, and he's the one that has transformed our work. So Etsuko has been with me for 10 years, and she does the topics that are relevant to the Institute. So each year she will take on a topic of a discovery at the Institute and transform that over the year into a preferably 2 to 3 minute animation. It's usually 3 or 4 minutes, but shorter is better if it's possible. Animation that shows how the discoveries, how a particular cancer works, or how type 1 diabetes works. She's currently working on P53, which is it's related to cancer and cancer therapies or cancer research. So, and it's fantastic. Actually, her P53 work's almost done. But Justin Muir comes from 20 years in the games industry. He worked on Mortal Kombat, the original one, and a few other things. He also worked for a couple of years in biomedical animation, so he understands what we're needing. But he came in with the programming skills and he's the one who's created our pipeline now, which it's a radical change for us, transformative for our possibilities, and it's really, I'm just going, we have to think bigger now, much bigger. And so that's really exciting, but I certainly know that there are a lot of other groups and institutes who are doing similar sorts of efforts, and I think we're all similarly excited about where the possibilities are going now. If you want to see any of our animations or request downloads, WeHi.tv is our main web portal. BioInteractive is the Howard Hughes Medical Institute web portal aimed at high school teachers and high schools, and they invest millions of dollars each year on outreach and all sorts of things. And they're a beacon in the world for science education. And then I'm reasonably busy on Twitter. I'm not a social media person, but just in the last 2 years I've got hooked on Twitter. And certainly if you want to know about when we publish all these new animations, or I follow a lot of microscopy. I follow a lot of special effects people and video game developers. So if you like that kind of stuff, that's what I tend to tweet about. And then science advisories, I'm always working with scientists who are experts. So Mike Kuiper, he works at the CSIRO in Melbourne, and he is a molecular simulation guy. He's amazing. I love him. He did the ATP molecule simulation at the very beginning I showed, the orange one, and he does proper real simulations where my work is, if you could see it, it would look something like this. It's an emulation of simulation. It's not really a simulation of molecules. And then Alistair Stewart, Victor Chang, Cardiac Institute, he's the expert on ATP synthase. So I think he's based in Sydney originally, but he's awesome. And then others from around the world are contributing various other things as well. So, but unfortunately, I was hoping to be able to show you more, but I can't of that current project, particularly the stuff I'm really excited by. But at least I think you get a taste of what we're up to. Because, and this is where I work. This is the Walter Eliza Hall Institute. This is the 6-story tall illuminarium that we installed 5 years ago to celebrate 100 years of the Institute. And it displays a non-stop roster of raw images from the microscopy in the Institute. And it acts as a beacon to draw people's eye from over at Melbourne University. They can see, standing at the tram stop, see the Institute and go, what the hell's going on over there? So, and actually I'm just we installed that, and it was only supposed to last maybe a year. It's lasted 5 years, but it's coming apart. And we're going to install a brand new one later this year. But with that, I will stop. Thank you.